All right, so today in class we talked about the cell membrane and how it is an integral part of the outside of a cell and allows things to move in and out. So we were just looking at the structure of the cell membrane today. And we're looking at the main character, which is a phospholipid. These phospholipids have two different parts to them. So one is the idea that they have a polar head. And this polar head is polar because it has positive and negative charges associated with it. The nonpolar tails are dangling off of that polar head region. And there's really no charge associated with these. They're sharing their electrons evenly. And it's just made up of carbon to hydrogen bonds. Whereas up at the top with the polar heads, we have phosphates that tend to have a positive charge and oxygen that tends to have a negative charge. So if we try to build the actual cell membrane itself, we're just going to stack some phospholipids in the way that they would form inside of a cell membrane. So notice that the polar heads are on the outside and the nonpolar tails are in the middle. And so next to that, we're going to put one of our proteins. Okay, and these are the channel proteins. So I'm just going to label this toward the bottom. So these channel proteins will be stacked next to the phospholipids, and they're just going to allow things to move in and out of the cell. So it's very similar to a door. Sometimes they even have like a little hinge on the bottom that would allow it to open and close. But these can actually move together or they can move apart to allow things to move in and out. Okay, so next to that, we'll draw a few more phospholipids. And the second type of protein that could be in a cell membrane is something called a receptor protein. So the receptor proteins have these little finger systems at the top. Okay, so here's our receptor protein. So this receptor protein only responds to chemical and electrical signals. So something could attach to the top of this that fits it just like a lock and a key. And then it's going to cause a change at the bottom of the protein so that something moves into the cell, but nothing will actually physically move through it. Okay, then we're going to add some more phospholipids just to separate these proteins from one another. And this next one really hangs off of the top of the cell, and this is called a marker protein. So this marker protein just helps to identify the cell. So it's like a name tag for the cell. So that basically like your stomach cells would stay with other stomach cells, your liver cells would stay with other liver cells, and that helps to form the tissues that make up those organs. If these didn't have identifying structures, then foreign invaders could easily attack the cell just because they wouldn't identify which one it was. Okay, and then finally, we're going to add some little clusters of dots in these nonpolar tail regions. Okay, and these are called cholesterol. So this is just what allows the cell to make the cell membrane kind of rigid if it's present, or it could be a little bit more flexible if it's not present. So the cell can kind of expand or it can contract just based on the addition of cholesterol. Okay, so all of these proteins and phospholipids have to match in their polarity and non-polarity. So basically we'll use a different color. But if we draw a line right down the middle of where these polar heads match the non-polar tails, we can see that these regions, like in this region, we have a polar head. So we would expect this part of the protein to have to be polar. The non-polar tails are in the middle, so we would expect this part of the protein to be non-polar. And then same thing down here, polar heads, so this part of the protein all the way down, these parts of the protein should be polar to match those polar head regions. Okay, so I'm going to post a picture for the virtual students of what we did as an assignment so that you can complete that and then get that submitted. Let me know if you have any questions.